Thank you all for coming. So our seminar speaker today is our very own postdoc, Dr. Lorna Kearns. Lorna got her BS with honors in geological sciences at the University of Leeds. Then she got her master's of research in marine geology and geophysics, everyone's favorite topic, at the University of Southampton. And then she got her PhD in paleoceanography and paleoclimate, also at the University of Southampton. Then she was the Joanna M. Reisig Research Fellow, a fellowship from the Cushman Foundation for Foraminifera Research, where she did about 6 months worth of postdoctoral research still at Southampton. And when that finished, she and her partner drove around the UK in a van for a couple months, which rocks. And then in a major step down, she came here. No, I'm kidding. But then she did come here for her postdoc starting just this August. And so we're very excited to have her here. And she's going to talk to us today about species evolutionary change and ecological structure change in the Middle Eocene during a time of major climate warmth. So with that, I will let you take it away.
So yeah, as Chris says, I'm quite new here, so I won't really be talking about my postdoc because I have no results yet. So so this will mainly cover 2 chapters of my PhD, and then it will also go into kind of a fun project that, as Chris mentioned, I got some funding for that is kind of carrying on as I'm here. So first of all, for those that don't know, forams are amazing. They are single-celled marine protists, and they're found in all global oceans, and they date back to around the Jurassic period. And they accumulate in really high numbers. So like a small vial will have tens of thousands in them, hopefully. So they make really good, they're really good for studying loads of different questions related to life and its relationship to climate. And as you can see, they've got a really wide range of morphologies, and they have a really wide range of sizes. So some range from around 50 microns, some all the way up to around 300 microns. So we can use them for a lot of different things: diversity, geochemistry, paleoceanography, and even questions of how individuals react to climate. So for my talk, I'm going to be covering around 3 of these, which is diversity, paleoceanography, and functional traits. Sorry. Thank you. So first of all, no. Oh, I see. Okay. So first of all, I'm going to be covering diversity, and this made up a huge amount of my PhD. So for this, I'm going to be focusing on the Middle Eocene period. So this is a period that's bookended by 2 climatically extreme events. So we have the early Eocene period, which is a greenhouse period. So the Earth had a really hot climate. And then we go all the way through to the EOT at around, I think, 34 million years ago. And this is when Antarctic ice sheets become permanent. So this is when we enter this icehouse world. And at this point, at the EOT, there's quite a lot of what we believe to be foram extinctions, or particularly a lowering of diversity. So that period of time has been covered quite a lot. However, this period here is known as the MECO. So the MECO is known as the Middle Eocene Climatic Optimum. It is a period of transient warming, and it's a 200 to 500 kiloyear event. And we think that sea surface temperature increased by about 3 to 6 degrees C globally. And a lot has been studied about this event in terms of paleoceanography and sometimes in terms of foram diversity. However, we don't know a lot about really how the forams reacted in this period here, which is when the most extreme cooling of the Eocene happened. And we also don't know how they reacted following the MECO. And this is mainly because I use ocean drill cores for this research, and there was a carbonate dissolution event during this period. So actually, there's not that many cores that represent the whole of the MECO and either side of it. However, for this, I'm going to be using International Ocean Drilling Programme, or IODP, Expedition 342. So this was drilled in the North Atlantic, and it recovered cores that covered pretty much the whole of the Middle Eocene. So 40 million years ago, it sat at about 30 degrees north. So it's not really polar, it is in a temperate paleolatitude. And this is really important to remember. So I really wanted to assess the diversity. And one of the things I was noticing was when I was looking at all of my forams, they were really small. And it was a bit unusual. They were a lot smaller than I would have thought they would have been for this paleolatitude. And there are certain protocols that we follow in foram analysis. And when we count diversity studies, we tend to cover those above 150 microns. And this is fine if we're in, the tropics. Everything tends to be larger at the tropics, so this is really okay to do. However, quite a lot of people tend to do this in the temperate regions, and we just don't know what we're missing when we do this. So a lot of my research was, figuring out what are we missing, what should we be measuring, rather than just following protocols. So when we measure at above 150 microns, we sometimes lose some of this diversity here of the smaller things. Some people may say that these smaller things are juveniles. They're young. They shouldn't be influencing our diversity count. But the problem is a lot of these species and genera don't actually grow that big. So we could just be missing quite a lot of information. So I figured out that if I was to use 150 microns, I would only be measuring the biggest of the biggest things in my, in my samples. And I'd be missing, all of this diversity. And that wasn't something I really wanted to do. But then measuring all these small things was going to take some time. And I actually wanted it to mean something rather than just measuring diversity. I really wanted to do this analysis to see, what are we actually missing as well. So I decided to measure at 2 size fractions. So everything above 63 microns and everything above 180 microns to really see what we were missing. So once I decided what I was going to measure, it was then how do we measure it? When we're measuring diversity, there are a lot of things you can do in ecology to measure this. And these are the 3 most common: species richness, Shannon's diversity, and Simpson's evenness. These are all great. I still think they're great, even after reading about them. But I think we can do better, particularly if we have a high number of individuals. My samples were made of thousands, and I can do a lot better than this. So this is what I got when I plotted, just a really rough diversity measure using these. And the problem with this is I can't really compare these lines. They're all in different units. They're all influenced by different things. So it's just a case of wiggle matching, and that's really not the best way to assess diversity. And there are other problems. All of these things are based on sample number. So if you only have one, or maybe you only have 10 to 20 individuals, that's really going to influence the diversity you get. I decided to count 300 individuals, so sample number is not a problem for me. Then we also have this key concept called the replication principle. So the replication principle states that when we have let's say we have this sample here, it's made of 4 unique individuals. When we add another sample with an equivalent 4 unique individuals, the addition of these should create, it should just be 4 plus 4 equals 8 for richness. However, this is not the case. So then when we do richness, we get this, we get this 8 species. However, when we do the Shannons and we do the Simpsons, they're not double what we were getting before. And this kind of breaks one of the fundamental laws of ecology. So this is why I decided to use something else. So in around the 1970s, someone decided to combine all of these measures together and create something called hill numbers. So it's a combination of all of these, so we can still get what everyone knows as a diversity measure out of them again. So if people are more comfortable, and as you'll see later, they're actually easier to present in presentations like this. So you can still get that. We're not limiting ourselves. So hill numbers rely on this thing here called Q. And this is what's important. So instead of having an individual number that represents diversity, we will get a sliding scale of diversity. So Q is a really important thing. So Q varies from 0 to 2. And in programs, you can set Q to be of any order you want. So it can calculate diversity at, I think I set it at 0.1. So as Q changes by 0.1 each time, a new diversity number is calculated. And from that, we get a curve instead of a singular number. So how this represents the normal diversity indices is, it's like looking down a microscope. So when Q is equivalent to 0, we see everything. It doesn't matter if it's abundant, doesn't matter if it's rare. This is essentially species richness. And then as we slide on to Q equals 1, we weight things according to abundance. So if something's rare, it's not having that big an influence on the diversity we get when Q equals 1. And this is essentially Shannon's diversity. And then we move to Q equals 2. Q equals 2, everything that's not common is just removed. We don't see them anymore. So when Q is equal to 2, we just see the common things that are making up those samples. Rarity no longer has an influence at all. And Q is calculated, in between all of these numbers, but these are just the integers that correlate to what we normally use. So Hill number will increase, and that is just an increasing diversity and the decreasing importance of rare taxa as we go along Q. So if we have a straight line, it means that it's an even assemblage. Nothing is rare, nothing is dominant, everything is represented equally within that sample. However, if we have a curve, that shows that here at Q equals 1, we have, Q equals 0, we have a lot of rare taxa, things that are not represented by that many individuals. And as we go down the curve, we remove those rare taxa until only common things are left. So here we maybe have one common thing that is dominating that sample, and that is an uneven assemblage. So when I did this, I counted at 2 size fractions, and I think I counted in total 22,300 individuals over the course of about a year. And that's a lot of data, and it took a very long time and it was very fiddly. But we got some amazing results from it. So these is just one representation. So I should say I never measured to species level. I can answer questions on that after the talk, but it was just a lot simpler to do genus level. And I also separate things into morpho groups, so grouping anything that looked morphologically similar together, and eco groups, so anything that lived in the same environment. And that was just a much simpler way of doing things for this time period. And the main thing I want you to see here is that here is the smaller size fraction. So this includes everything. And as we can see, the gradients of these curves are not as large compared to these, the more than 180 size fraction. So within this larger size fraction, we're seeing a lot more rare things because this curve is at such a high gradient that they're dominated by just a few big things. So like I said, I still believe that, the Shannon's diversity and richness is a good way to present this data. So here we have the climate at the time. So here we have this middle Eocene climatic optimum. And this is generic richness. And as you can see, kind of at the same time of this warming event, we have in the smaller size fraction a real increase in the amount of genera that are within my samples. Whereas the more than 180 size fraction, we don't really see this increase. We see a slight decline, and that is happening prior to the MECO. And this is when that cooling is at its most prominent during this period. And then we see this massive decline in the smaller size fraction, and this is when cooling occurs after the MECO. And what's causing this increase in richness is real increases in these smaller taxa. So we have taxa such as Chyloglobulina and Jenkinsina, which are just really taking over the assemblages at this point, particularly as other things are being removed. So a lot of the big things and the smaller photosymbiotic forams are being removed at this point, and they're being replaced, but not just replaced, they're being overtaken by these smaller genera. So when we look at Shannon's diversity, we see a slightly different effect. So once we take away all these rare taxa at the Shannon's diversity, we see that the smaller size fraction doesn't really, the MECO, which is happening around here, is not really having an effect on the diversity that we see in the smaller size fraction. However, the more than 180 size fraction is seeing this huge decline. But interestingly, it's not happening during this warming. It's actually happening during the cooling event prior to the warming. So this is really interesting. It means that the bigger things are actually being removed by cooling, not warming, and they're just not recovering after that. It's not the MECO and it's not this hypothermal event that's affecting diversity. It's actually the cooling prior to this event. And we see the same pattern happens in Simpson's index. And we see again, it's happening prior to the MECO and it's this big decline. Whereas when we measure everything in the more than 63 size fraction, we see that, you know, it's not really changing. It's kind of balancing itself. The community is reassembling itself to adapt to these changes. And just to show that this pattern isn't unique to genera, when we look at morpho groups, so we link everything related to its morphology, we see exactly the same pattern. We see that richness, you know, is a bit skewed because the smaller things become to bloom around this period, so we get a lot more. But we see this same pattern of the larger things are declining in diversity prior to the MECO. So from that study, we found that diversity dynamics in planktonic foraminifera are influenced by the size fraction we choose to analyze at. If I'd have chosen to analyze at the normal size fraction that's stated in a lot of papers, I would have lost so much diversity, and I would have said that the MECO had this huge effect on diversity dynamics at this time. And we found that larger foraminifera respond earlier, and that response is exaggerated compared to the smaller size fraction. The smaller things, because we measure everything, the smaller ones are coming in to compensate for those larger things being lost. And if you want to read more about that, I have a paper. So now I'll move on to my second chapter, which is more to do with paleoceanography, but it really did follow on from that first one. So when we looked at what was causing these diversity dynamics, we noticed that there's this one species, this shown in like this turquoisey green, that was not, it was actually increasing its numbers, and it didn't really have an effect from, you know, the cooling, the warming. It just survived and was quite happy with what was happening. And this genera is Subatina. Subatina is, you know, it's quite a boring foram to study. It looks quite boring. It's 3 chambers. It looks like a piece of popcorn. It's, you know, it lives in the thermocline. It's not exciting. Everyone who studies the MECO loves to study the surface ocean. They're the things that go extinct. They're the things that have like these ornate shells. But no one really wanted to study this, and I thought, oh well, let's give it a shot. Let's see why it's surviving. So for those who don't know, we can use forams we can use stable oxygen isotopes and carbon isotopes to really see where forams are living within the water column. And this can also tell us about paleoceanography at the time. So here we have this foram here, which lives in the mixed layer. And then we have Subatina, which is in the thermocline. And then we have this one here, which is in the subthermocline. So I picked out these genera. So this is Glabidrina theca, this is Subatina, and this is Catapsidrax. And we picked those out, and then we can see, you know, they're all stratified within this water column. So how did these things change through this warming interval? And this is what we got. So this is oxygen isotopes. So up here, well, you know, these blue ones here are the surface ocean. And these, you know, this is the thermocline and this is the subthermocline. And this is how we can see that it's changing. And so here we have the warming interval that happens here. So prior to the MECO, we see the subthermocline and thermocline dwellers are, you know, showing this oxygen isotope signal that indicates that they're living in cooler environments than they were at the beginning of my study interval. And this represents around a 2°C cooling event. And this matches globally with what we're seeing as well. The thermocline is really cooling, and so is the subthermocline compared to the surface ocean. As you can see, the surface ocean is, you know, it's still cooling slightly, but not as much as those in the thermocline and subthermocline. And then when we reach the MECO event, so this transient warming event, everything just shifts up. The oxygen isotope signals shift up till they're pretty much showing the same signal. And this is really strange because we would have thought that the surface ocean would warm the most because that is what the global signals show. However, my site was in the northern hemisphere, North Atlantic, and no one has really studied that area before in terms of doing this. And we found that, everything moved upwards, and the lack of warming we see from this surface dweller, this Globodrina theca, we actually have seen in other cores. So this is not unusual. What is unusual is a huge degree of warming in the thermocline and the subthermocline, pretty much about 7 degrees C. That's a massive amount to happen in, this happened over a 1 million year time frame. That's a huge warming event. However, we maybe think that our signal is being influenced by something else. We don't believe it's a full 7 degrees because that just seems extreme. But another study in this area did find around a 4 to 5 degrees C warming in the thermocline and subthermocline. So we can say that although the magnitude of warming is maybe a bit exaggerated here, we do still think we did have this major warming event. And this created, quite a strong, everything was living within the same depth habitat, really, according to this. And then following the MECO, everything returns to about normal. Everything here, okay, everything's cooled, but this is a global cooling event that happens over the whole of the Eocene period. But, the way the surface dwellers are sitting in the surface, then we have thermocline, then we have subthermocline. It's what we would expect. And the recovery is remarkably quick. For something that happened this extreme, for it to be back to normal within 2 million years, that's quite an impressive return. We see a little bit of, they don't fall back into place quite as smoothly. We see here that the subthermocline is giving a signal of above a thermocline dweller. But again, these are based on, just one vial of 4M. So we can't say if this might just be an odd sample. But we think this pattern is true. We just think that it's maybe exaggerated by a few different things. So that's what we found from these bulk isotopes, where we group loads of different individuals together. One of the things that's coming more and more common in foraminifera is that we measure individuals. So a lot of people don't like measuring individuals, the reason is it creates a lot of noise, and it's really, really hard to pick out these signals. But I wanted to measure individuals of subatina to see, if we can see why they're surviving during this period. So the pink dots here all represent individuals of subatina. And what we can see is, firstly, the gray of the subatina is always sitting pretty much in the middle of all these pink dots. So that tells us that grouping them all together does get a good signal of what all the individuals are showing, which is great. But what we do see that's really, really strange is we have this huge spread of data from these individuals. So some of them here, they're above the surface dwellers, and they're also below the subthermocline dwellers. So this is a huge ecological niche that these things are living in. And it isn't just at one time period. They have this huge spread in oxygen isotopes and in carbon isotopes, at every single sample I looked at. And I believe that this is why they're able to survive these really huge climatic transitions, is because they have such a wide ecological niche that when warming happens, it's like, well, okay, we have a population that's living in a warm area already, so they can survive. When cooling happens, oh, well, we're already living, below the thermocline, so we can survive that as well. So I think this is one of the real benefits of measuring individuals. We never really knew why they survived. It was always said that, they're generalists, so they will survive, but we didn't know what made them generalist. And it's this, I believe it's this, this ecological niche that they're able to live in such a broad range of environments. So another thing we did was measure the morphology of these subatinas in the hope that we could find a relationship between size and temperature. So when the temperature decreased, we'd hope they'd get smaller, and when it increased, maybe they'd get bigger, because we see that in forams anyway. So as you go towards the poles, they tend to get smaller, and as the tropics, they tend to get bigger. So we were hoping that maybe we could see this pattern across my samples in, this significant warming and cooling period. However, we didn't really see anything. We saw a slight decrease here in test size and a slight decrease in aspect ratio. So they were getting smaller and tighter, but statistically, this meant nothing. It didn't really show up on our statistics. So actually, another thing that's helping them survive is that their morphologies just don't change. They're happy to stay at a consistent size and in a really large environment. They do have quite a wide spread like they did in the stable isotopes. And again, we think this is helping them survive during this period. So from this, we found that the MECO caused substantial changes to the water column, and they were all concentrated within the thermocline compared to the surface ocean. And that we believe that subaquina survival was aided by trait plasticity and a generic lifestyle. And we think that more individual forum analysis should be done to understand if this is what's happening in other groups. If by dissolving everything together, are we missing this huge amount of ecological information? And again, I have a paper on this if you would like to read more about that. So I've got quite a bit of time left, so I can go through this called functional traits. So this is a brand new topic. I got the data last week, so it's all a little bit new, but I think it's really exciting. So a functional trait, the idea being that when we measure a species or a genus or anything, we want to be able to measure something that is consistent across all species and even across, across different animals, across different plants, so that we can find this thing that we can measure consistently throughout the animal and plant kingdom that reacts to an environmental variable, mainly temperature. And the way we do this is we find a trait that we can easily measure, so something morphology wise, and we find an environmental variable, and we'd like to see this correlation. And if this correlation exists, then I no longer need to look at the species of the forams. I can just measure this trait and see how it's reacting to climate. Makes my life a lot easier, and it's really interesting. However, we tried to do that in the previous paper, and we chose size because size is really easily measurable. We know it has some relationship with temperature, and it didn't work. We didn't see any relationship, and that was really annoying because size is so easy, and it would have been great if it would have worked, but it didn't. So then we thought, okay, well, we need to move on to something maybe more complex. And for this, we chose porosity. So go on to this one. These little holes in the forams are called pores, and we can measure those. Those holes, and we think, and we know from studying surface dwelling forams that have symbionts, that porosity is related to temperature. As we increase temperature, so porosity also increases. So we have this really nice relationship. And we think these pores are important because we think they facilitate gas exchange of the foram. We've seen it in benthic foraminifera, where mitochondria cluster around these pores. So we think that they are in some relationship with gas exchange. And we think that porosity increases with temperature because as temperature increases, the forams need more energy and they therefore need more oxygen. So they need to increase their porosity. So this should work. But we know this exists for surface dwellers, but the problem with surface dwellers is they have these symbionts, which are interacting with all of these problems, like with porosity, with the way they grow their shells. And the problem with that is we can't unpick the effects of these symbionts. So we really wanted to do this on some thermocline dwellers which don't have symbionts to see if this relationship upholds. So I picked these two species. So the reason I picked these two species is that they are descended from each other. So the first one is Spheroidinella opsis paneidehensis. And the one that's descended from it is this Spheroidinella dehensis. They look quite similar, but scientists believe that they are different genera, not even just different species, they're completely split. However, we find them in the same samples and one increases while the other decreases in abundance. And they look so similar, like how are you meant to pick them apart? And the way we pick them apart is, so this one here has this one big opening called the aperture, whereas this one has a big opening and a secondary aperture. The problem is this secondary aperture is so small in some of them that you can't even see it. So for around, I think it's around maybe 1 million years, you can't really tell them apart. This split may have happened way earlier. I should say these are from the Indian Ocean around, I think around 6 million years ago. So I only did one sample because what I was going to do was going to be really costly and I only had 6 months. But the way we measure the pores, as you can see here, like some of them have pores, some of them don't. But the way we can measure them is to get inside the test and measure them internally. And that's what I decided to do. We decided to only measure this final chamber because we know that porosity changes as the foram grows. So the final chamber should show us its true porosity in its final environment. And the reason we chose these two species as well is because we can use porosity to tell apart species. So in theory, if they are different species, they should have a separate porosity. So to do this, I loaded them all into drinking straws with a little bit of botanical foam in between each one. And I did around 50 of each one. And we sent them off to a micro CT scanner. And we come back with these amazing slices, which are cut through the foram at regular intervals. And then what we do is we get that into a big program, and then I go through each slice and mark up the final chamber, which is seen here in green, and I mark up the pore, which is in pink. So this, in the end, will give us a 3D representation of the pore. No one's ever done this before. We tend to just measure, internally an aspect ratio or just the size of the opening. So I was measuring 3D. Now, each of these forams comes with about maybe 400 to 900 slices. And I have to go through each single one and measure it up for 100 individuals. So as you can imagine, this is a really time-consuming process. But in the end, we are left with like this amazing image. This is a 3D representation of the pores within this one foram. And I can't show it today because I don't think it'd work on this computer, but I can generate 3D spinning models of, we can see the pores within this final chamber. And I think this is really cool because we can see, different sizes, and then I can go through and measure, each one of these pores is measured separately. So I can get loads of data just from each individual pore. And normally when these studies are done, you only measure maybe 10 pores because it's so time consuming. But because we're doing it in 3D, I can measure every single pore within this final chamber. So the amount of data you can get just from one individual foram is amazing. And the thing is, I have these scans now forever. I can go back and measure, other chambers to see how it changed through time and generate more 3D models. So what we found was that porosity is indeed different between these two species. So this is the earlier one, and this is the one that's descended from it. And we can see that porosity is increasing massively. So this has an average porosity of around 9. This have an average porosity of around, I think it's 4. 4 to 9 may not seem that big a difference, but for a foram, that's huge. The max you can get is around 20, but that's really, really rare. They're sitting within the normal bounds we'd expect, but we wouldn't expect this to happen within the same depth habitat, in the same location, in the same sample, if they were not separate species. So I think from this, we can conclude that, they are separate. Maybe it'd be really interesting to go back from when we think they split to see if this porosity does increase like over time. And that would be a really interesting thing to do. So then I was interested in, okay, but what is causing, how is porosity increasing? How is it doing it? Is it getting bigger pores? Is it getting bigger, more pores, but smaller? And because the thing to note is that these are substantially bigger forams. These are they're maybe double the size of these. So I was really interested to see like how are they managing to cope with this size increase? So then I can get mean pore volume. So that is out of all these pores, what is like the mean pore per foram? And then how does that relate to porosity? And what we can see here is the one that's descended from the other, the newer one. It has much larger mean pore volume. And therefore, that's what's giving it its more porosity. But we can see that they are kind of clustered. It's actually just that this is like, these are consistently having more porosity than you know, the ancestor one. And then we can look at the number of pores per micron. So this is like this is a size normalized count of how many pores were actually in that final chamber. And what we can see here is that the descendant has a higher pore volume, but it actually has lesser number of pores. So the way this is increasing its porosity is that it's actually decreasing the number of pores, but it's getting bigger pores. So it's taking away the abundance of pores, but they're just getting bigger. And then what we did is a really cool tactic we can use is we can use magnesium and calcium to understand how the temperature that they were living at. Oxygen isotopes can be used for temperature, but they have a few flaws and a few different things that can affect them. Whereas I like to use magnesium and calcium. So I got a laser and I put some holes into this final chamber and measured the magnesium and calcium 3 times over each final chamber to generate a record of temperature or magnesium and calcium. And this is what we found. So as magnesium and calcium increases, it's a higher temperature. And as we can see here, there is a relationship. So we get this, the ones that have this higher porosity tend to have a lower magnesium and calcium. And then the ones that, the ancestor, they're pretty consistent. There's no real change. Statistically, there's no change no relationship between porosity and magnesium and calcium. But we do see this relationship in this descendant. So that's really interesting because this is not the result that we really wanted. Ideally, if it was going to follow the same thing as the forams that live in the surface ocean, we would have got like this nice straight line here. Whereas what we're seeing is that this functional relationship that we see in surface dwellers does not correspond to those in the thermocline. And we're seeing that from these results, although this descendant is bigger and has a higher porosity, which should mean that it's living in warmer waters, we're actually seeing the biggest and the most porous living in even deeper waters, even cooler waters. And this is in the thermocline, so you know there isn't that much change, but they are, I think, from my memory, it's about a 4-degree difference between some of these forams. So the average, there is a statistical difference in the magnesium and calcium, and it's around 2 to 4 degrees between this one and this one. That's a huge change within the water column. So if the magnesium and calcium is giving us a true signal, that means that not only are they different species and different genera, they're always living in a different environment. And it also means that, unfortunately, porosity does not relate to temperature in a nice, easy way so that we can measure porosity in the future. But it does show that forams are really complex, and we can't take what happens in the surface ocean as, you know, as the book. We can't say that that applies to everything that we measure. So there's new data being added to this pretty much every day, but hopefully I think it's already something cool, but maybe these relationships will get even stronger. I think I have about 20 more to add from each, so hopefully that'll get even stronger. So from that, we found that porosity does indeed vary between these genera, and it varies in a way that is consistent with having them being different species, but it does not vary in the same way we would have expected. And porosity does not have the same relationship with temperature that has been observed in symbiont-bearing planktic foraminifera. Therefore, the hunt for this perfect functional trait that we can measure across all forams is still ongoing, unfortunately. But I think it's a really interesting avenue, and I think we will get there. But functional traits were developed on plants and leaves, and they are, you know, they're complex in themselves. But the problem with forams is we can only observe them in life, you know, days, if that, and we can't see what's happening inside them. So we're having to take these observations and assume that they're the same in the past. And that is really difficult to unpick all of these abiotic and biotic variables that are happening. So thank you for listening. Are there any questions?
We have plenty of time for questions, and Constantino is readying the questions cube. So as soon as he's ready, raise your hand.
It's nice talk. So you got these climate or whatever environment driven excursion. I just wonder, when you do the analysis on times that are kind of boring in Earth history, what is sort of the reference level of fluctuations in these metrics that you look at, and what's sort of happening as a background evolutionary trend? Right. So if you do all of these detailed things, what happens in the places where nothing should happen?
Yeah, so I think that's the problem, is that when we look at these forams, we assume that something that's happening, in the North Atlantic is happening in the South Atlantic. But unfortunately, that's just not the case. Each of these, the signals that I was getting in terms of diversity, I do believe are just these regional signals. And I think that's the problem, is that we don't look, we tend to just only focus on maybe the tropics because they're interesting. And then we don't have these background, signals to actually look at. Luckily, we do have that every expedition that goes out does create these stable isotope curves. So we do know the climate pretty well in each of these things. But the problem is that we just need to do more of this foram analysis in these background intervals, because I think the background intervals are really interesting, because that's when species are recovering, and that's when, some of them are like going towards extinction within these periods. So yeah, I think more we do have the climatic background, but it's just, getting this diversity background is hard work.
Speaking of hard work, if I may, I really hate machine learning, but is this maybe a place where you want to try it? I mean,
yeah. So this was part of a group in Southampton called Piston, and it was a big scale project, and they are machine learning how to extract. They're extracting chamber size through ontogeny, so as it grows. I just wanted to do something a bit different and do the pores. The pores are slightly more complex. I think it took us 5 years to get the machine learning of, the chamber sizes down. So this would take a lot longer, but it's ongoing. Yeah, it is something we're looking into.
Great talk. I'm curious about how you differentiate whether a population has died out and then, evolved to live in a different part of the water column, as opposed to, how do you differentiate the climate signature from the change of habitat? Or do you kind of end up in a circular loop? So that particular plot that you showed with the 7 degrees C warming, how do we know for sure that that's a temperature signature as opposed to just a change in where they lived in that same time window?
Yeah, so that's a really important point, is that we know that because other studies they've studied other subthermocline dwellers and other thermocline dwellers. So we know that they're not moving. For these forams to move within the water column, it would require such changes to their ecology that we just don't think it's happening. And other studies in other areas that have measured multiple species within, the thermocline, they all follow the same climatic signal. So we can just, all of these groups, and it's really just unlikely that, 20 species are moving within the water column.
Hi, Lorna.
Hi.
Adrenaline drop first. Really good talk, really awesome. I've got 2 questions. One of them kind of like builds on Sean's question, and that is, you see these kind of changes of these larger species across your interval, and do you think that's because they're migrating because of the changes globally, and because obviously you're in the northern hemisphere, are they moving polewards or are they moving equatorwards? What's going on?
So I wouldn't say there could be migration. I will say that there could be migration. During the MECO, we actually see the biggest effects more on the northern poles. So if they were going to migrate up there, I wouldn't have said that was where they were going to migrate. I would say they'd migrate towards the equator. So yeah, they could be migrating. I think it's really hard to pick up that migration signal unless you do multiple cores along a transect. But yeah, they could be. The fact that we still see them there, and they're decreasing in abundance, but they're still there, and they're still there in like quite good numbers. Just because I only measure 300, there's still vials full of them. So I think they could be migrating, but I'd maybe need to go, more samples in between those intervals to really see if they were dropping off, at a consistent rate, or whether they're just like, because subatina does drop off slightly, but then it comes back. So I doubt it would have migrated and then come back.
Okay, and my second question is, now that you know what you know now about the flexibility of species like the genera such as subatina and the spheroidnalopsids in the kind of more modern ocean. Hang on, I just forgot my question. That's changes that you see across the MECO, where the kind of this thermocline, subthermocline, and the surface dwellers are all kind of occupying the same habitat.
Yeah.
Around that period is when a lot of mixed layer species like Acarinina die out.
Yeah.
Around that kind of 40 to 38 million year time period. So you show that there's very little change within the temperature in the surface mixed layer.
Yeah.
So if it's potentially not a change in temperature, what do you think might be causing those species to die?
So I think what's causing those species to die is the previous cooling. I think the cooling from the beginning of the Eocene really affected them. And then I think we have this, I think it's like a 2 million year period before the MECO, when cooling is at its most rapid. And I think that is what really caused these things to decline. If I was to do, a species analysis, I think I would see that the number of species represented in like Acarinina or Morosavella, they do start to decrease. I wasn't finding that many of them. And, they weren't very big, which I'd usually expect. They were really tiny, and they just never really recovered. So I think that MECO signal that we're seeing is actually a previous signal. I think it's the cooling, not the MECO itself.
Cool, yeah.
Thanks for your talk. I was wondering, I thought it was really cool how you showed the hill numbers were this sort of continuous measure of diversity across that axis. And that contrasts with doing like two separate size fractions that are like, just have this sort of well, I don't know if it's arbitrary. So I guess my question is, how do you define what that cutoff is for a size fraction? And do you think it would be possible to also do like a continuous look at diversity, continuous both in like Q space and like size space?
Oh, that's really interesting. Yeah, so I think in terms of the size space, I think you could, if you measured like really small size fractions, you could just add another axis to those graphs, and you could get like this 3D view. The way that we determine the size fractions is really, it's just a standard protocol. The reason I picked more than 63 and more than 180 is not really used that much in foram analysis. The reason I chose that was to almost exaggerate this difference, because more than 150 would have maybe incorporated a few small things, and I don't think it would have shown as good an image as what we got. But I also think that the more than 180, from looking at the sample, they were all on 180. Doing more than 150 wouldn't have been valuable. And I think the main point that I wanted to come from that paper and doing that study is that we should be looking at our sample and choosing it based on the sample, not just, oh, the standard is this, or maybe we should just do this. It's like looking at what you have and saying we should measure, related to what's in there.
Thank you.
Hi, this might be a dumb question, but how long do foraminifera live?
It's a great question. It can be anything from, I think the average is around 3 weeks. That's, some of them live months, but some of them live days. The ones that I were picking were all adults, so I would have assumed they'd live 2 to 3 weeks.
Okay, cool.
Great talk. Thank you very much. I don't know much about like foram structure and also like features of it. You mentioned that you looked at like porosity size and like the chamber size and just the general like size. I'm wondering like what other features would you be interested in researching to develop like the relationships that you're interested within like foram analysis?
So I think, when I've done another study that I didn't talk about here, and I measured around 15 variables on, the foram. I did, it's actually not the best one to do it on, but I did aspect ratio, which is across here, aspect ratio here. Each chamber individually was measured for size. Porosity was the one that's like most probably that combination of being relatively easy to measure, but still quite interesting. I really wanted size to work because it's easy. The aperture, I think, is like a really interesting thing that no one really measures. It's this huge opening and they change in size, and it's one of the ways we identify species is to look at the size and shape of the aperture, but no one really has studied that. I think it's this, what I'd want to see going forward is this combination of different geochemical techniques combined with the morphology. Because what we found in another study was that we measured all of these 15 variables, and I was picking apart these species by eye. But when we looked at all these morphologies and tried to do a principal component analysis to see if all of those 15 variables could pick apart these species, it just couldn't. And, you know, I'm seeing it with my eyes, so I know they're different, but it's just what we're measuring that's not picking it up. But I think the developments in micro CT scanning so that we can start to measure these, you know, measure these 3D traits, I think is going to be the next step.
You were showing these CT scans. I was wondering, sort of, like, how close do you actually want to look, right? Because in the end, I guess you are in some sense interested in average properties, right? And so, so there's this issue. You have a really detailed representation of one exemplar of a big sort of ensemble, and I'm sure there's something to be done there. But then I was also wondering, so you found that somehow porosity increases, but it goes by like bigger pore space or the other way around or something like that. And so evolutionary, there's presumably some functional optimum there where these things trying to flux things through as much as possible. And then they want to have high surface area. And then there's some sweet spot sort of in between. And is that something that you can take and then see how that adjusts given on what have you, nutrients or something like that?
Yeah, so one of the things that the problem with the machine learning aspect is it needs data. So I'm hoping this is going to be the data, and then we won't have to measure, you know, each individual pore within the final chamber. We'll be able to measure maybe a quarter of the final chamber. And from that, we'll also be able to then measure more individuals. So yeah, I think they do flex. So like, you know, they'll have loads of pores, which are really small, and then they'll go to having loads of pores, which are big. And they'll just keep adapting until they, you know, they meet that one sweet spot, as you said. And I think that that'd be a really interesting study. And it's one thing I would really like to do is to track this lineage from when we think they split to like 2 million years afterwards. And I think we would see that, you know, the pore sizes would change and then come back smaller and then go bigger. And there'd be more pores and then less pores. And then as we begin to see them as morphologically different species, that is when they'd settle into quite a consistent porosity. And that's when I think we can start measuring, you know, not every pore within the chamber.
Yeah, that might be a silly question, but they're also limited by the food they can have, the carbon, you know, and so during this event, the Miko event, they all move to a layer where there is more food, basically. And so, you know, you also spoke about this carbon dissolution events, stuff like that. So could the amount of carbon available in the ocean, you know, play a big role on this big change that you see, not only, you know, temperature and stuff like that?
Yeah, so I did look at the carbon isotopes, and we did see similar changes, but they're just, the problem with carbon isotopes is they're really hard to unpick what's actually happening. We did see the gradient between all the species did change at the Miko, and we interpreted that as a decrease in primary productivity. However, in another ocean, they show an increase in productivity, and it's really hard to unpick what's actually happening.
Also, over time through the Eocene, this carbon seems to decrease because when you reach the Oligocene, there's these diatom blooms, they're silica based, stuff like that. So you have an overall decrease of all the time, the carbon. So that may play also a big role.
Yeah, so I think this is important to look at, but I think it requires loads of micropaleontologists looking at different microfossils to really see what's happening. That's actually something I'm going to be doing my postdoc in after the Cretaceous extinction. So yeah.
Anybody else? I'm glad we had so much time for questions. That was awesome. There's a question in the chat. Oh, does anybody in the chat have any questions? While you're bravely considering whether to raise your hand or not, I have a question too. It's kind of following up onto Adam's. So if it was cooling prior to the MECO that really drove these extinctions, what is it about cooling that would have done that? Because it's the middle Eocene, so it went from like 35 to 33, right? Like it was hot to slightly less hot. So what aspects of the ocean changed with that cooling that would have actually caused?
So I think the things that we were losing during that time were like Acarinina, Rosavelids. They're the things that have symbionts. And I think it's that relationship between the foram and the symbiont, which was actually changing. And I think the symbionts did not like that cooling. They'd been at a consistent temperature, and 2 degrees for these symbionts, I think, would have been quite a huge change. And I think some of them would have been able to, hang on, trait plasticity within them. But I don't think the whole population would have been able to hang on. And then once a population decreases to a certain amount, they just can't recover from what's happening. And then I think actually the MECO maybe was then too warm. It was too much of a change. It was like cooling and then a warming, that any that survived that initial cooling were then just shocked by the initial warming.
This is a really cool talk, and I'm like brimming with thoughts and questions. It's really interesting. Thank you. Final call. No? Okay. Thank you all for coming. I believe we're going to get beers at 4. I mean, Sean and I at least have talked about going to get beers at 4. So, and Lorna's going to come. So if you'd like to talk more about this, Austin Beer Works, 4